So howdy, my name is Natalie Chavez,
I'm Mayra Landeros. I'm Sofia Guerra.
and today we'll be presenting summary of Chapter 3 and Chapter 4 for immunology.
What is an antigen? An antigen is any substance that can induce an immune response, specifically by binding to an antibody or receptor on a B or T lymphocyte. Antigens can be proteins, polysaccharides, lipids, or nucleic acids. Here in this image, we're able to see where we have different binding sites depending on whether it be B or T cells, but here we are presenting a B cell. Moving on, immunogen. They induce an immune response, while haptens are small molecules that require a carrier protein to be immunogenic, and foreign or self-antigens originate from pathogens or self-molecules. What is an epitope? An epitope or antigenic determinant is the specific part of an antigen that is recognized by an immune receptor, such as an antibody or a T cell receptor. Types of epitopes are linear epitopes, a continuous sequence of amino acids. Conformational epitopes are dependent on the 3D structure, and antigens have multiple epitopes, while dominant epitopes induce a strong immune response. And here we can see an image of where the epitope takes place, where the Fab region is, and where the antigen is binding onto the epitope site. T lymphocytes. They recognize processed peptide antigens presented by major histocompatibility complex MHC molecules on antigen-presenting cells. CD4 plus T cells, helper T cells, recognize antigens presented by MHC class 2, extracellular antigens. CD8 plus T cells, cytotoxic T cells, recognize antigens presented by MHC class 1, intracellular antigens. B lymphocytes. They recognize native antigens without the need for antigen processing. The receptors, called B cell receptors, BCR, can bind directly to proteins, polysaccharides, lipids, nucleic acids in their natural forms. Upon activation, B cells produce antibodies that neutralize pathogens.
Moving on to the major histocompatibility complex, or MHC molecules, and their classes. MHC class 1 is found on the surface of all nucleated cells. It presents intracellular antigens to CD8 plus receptors on T cytotoxic T cells. The antigens are peptides, indicating the assistance in killing a virus-infected or cancerous cell. MHC class 2 is found on APCs, or antigen-presenting cells, like dendritic cells. MHC class 2 presents a variety of extracellular antigens of many types of macromolecules to CD4 plus receptors on T helper cells. T helper cells will then coordinate an immune response. Moving on to the antigen processing pathways of MHC class 1 and MHC class 2, there are two antigen presenting pathways respective to class 1 and class 2 MHC. Class 1 MHC, as mentioned previously, processes cytotoxic proteins or intracellular proteins using a proteasome transporting the peptides to the endoplasmic reticulum with the TAP molecule to load them onto MHC class 1. In MHC class 2 pathways, it processes extracellular antigens, which are taken in via phagocytosis or endocytosis, then is degraded by lysosomes and placed on MHC class 2 for presentation. What is the role of dendritic cells in capturing and presenting antigens to T cells? Immature dendritic cells capture antigens via phagocytosis and endocytosis via the activation of toll-like receptors or cytokines like TNF and IL-1. The dendritic cells will then migrate to the lymph nodes and mature. Mature dendritic cells will present their antigens to MHC class 1 to CD8 plus receptors on T cytotoxic T cells, and MHC class 2 to CD4 plus receptors on T helper cells. This activates naive T cells. This also allows extracellular antigens to be presented to T cytotoxic cells by expressing it on APC's MHC class 1. This process is known as cross-priming or cross-presentation. This helps in the activation of T cytotoxic T cells when the antigens are not present intercellularly, although dendritic cells present both MHC class 1 and MHC Class 2, this is not cross-presentation. Cross-presentation or priming is a redirection of extracellular antigens to be presented by MHC class 1. Moving on to Chapter 4, antigen recognition in the adaptive immune system.
Development of lymphocytes. The development of lymphocytes begins in the bone marrow where hematopoietic progenitors commit to either the B or T cell lineage. B lymphocytes mature in the bone marrow, and T lymphocytes originate in the bone marrow but migrate to the thymus for maturation. Although the final maturation between B and T cells differ, lymphocyte development always begins in the bone marrow.
What types of selections take place during different stages of maturation? In the thymus, the maturation or development of T cells can be negatively selected where a cell has a strong affinity to bind to self-antigens. This kills the T cell, preventing autoimmunity. T cells that are positively selected can recognize self-MHC molecules, and this selection ensures that T cells can interact with the host's MHC molecules. B cells also undergo a variety of similar selections, but this takes place in the bone marrow.
Negative selection during maturation. Negative selection removes self-reactive T cells to prevent autoimmunity and promotes immune tolerance, also known as apoptosis. It ensures that only T cells with appropriate antigen recognition survive. On the other hand, those that harm the body are either eliminated or converted into regulatory T cells. Receptor diversity and adaptive immunity. The vast diversity of BCR and TCR in the lymphocyte repertoire is generated through a combination of genetic mechanisms that allow the immune system to recognize a limitless number of antigens. Somatic recombination, also known as VDJ recombination, generated by recombination of variable V, diversity D, and joining J gene segments. Junctional diversity, random addition of or deletion of nucleotides at the junctions between V, D, and J segments by enzymes like TDT. Combinatorial diversity, different combinations of VD and J segments create numerous receptor possibilities in B cells. Pairing of different heavy and light chains further increases diversity. In T cells, different alpha and beta chains add another level of variation.
Thanks, and these are our resources. Okay.